Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today I will talk about basics of CRISPR and we will also learn how to design guide RNA or gRNA in CRISPR experiment. CRISPR-Cas9 is a popular tool for gene editing and one of the top line gene editing tools. It relies basically on two main components as it is indicated in its name CRISPR and Cas9. CRISPR actually stands for Clustered Regulatory Interspaced Palindromic Repeats, while Cas9 stands for CRISPR associated protein 9, which is actually an endonuclease that allows double breaks or double strand breaks, or you can say that that has the ability to cut both strands of the DNA. So this is the whole theme of the CRISPR, it uses an endonuclease for cutting, and the technology is very specific, and it can cut or insert or delete a specific section or a specific gene, whatever you call it. Okay, so just we will talk about slightly for the introduction or for the working procedure of the CRISPR, then we will move to the tutorial section quickly. Okay, let's say this is our DNA, and if you are interested in this region to cut, so we will call it the target region. But in CRISPR, there is another concept, we call it photospacer adjacent motif, or also we can call it PAM. PAM is actually the combination of two, three to five nucleotides or base pairs, and it is important that if this is your target region, then in the upstream position of your target region, you have to find out the PAM nucleotides, or those three to five nucleotides, or those three to five base pairs. So, what is the role of PAM in CRISPR? PAM actually helps to recognize the location of your target DNA which you want to cut. Okay, how it recognize the endonuclease enzyme? It recognize those three to five nucleotides or base pairs, which we call it PAM. So to conclude this slide, if this is your target sequence or this is your target area, so you must first identify a PAM region in the upstream region, in the upstream location. From the upstream, I mean 5 prime location. So once you identify the PAM region, then we will design, or you will use your primers. What are actually the primers? In normal PCR, we are using forward and reverse primers, but in CRISPR, the concept of primer is attributed to the guide RNA. So once the nuclease binds here on the PAM location, or on the PAM base pairs, then the guide RNA will guide the nuclease enzyme where to cut that specific region. So, in other words, we design the guide RNA or the gRNA before launching an CRISPR experiment. So now we are going to cover some interesting tutorials. In this tutorial, we will learn how to fetch a gene from the database itself quickly, and then how to find out the PAM sequences, and then how to design the gRNAs, and then to calculate some efficiency analysis for gRNAs. And we will complete this task using Google Colab. So let's do it. So I have this browser. You have to write Google, sorry, colab.research, google.com / github /h100. If you don't know, or then simply it's better to write Google Colab and then click here, welcome to Google Colab. Once you click here, then click here GitHub and now write ASH100. This is my repository and hit enter. So you will see this kind of thing. Now click here and search for primer designing. Click here. So you will see two files. This file CRISPR gRNA design. So click here. Once you click here, you will reach to the respective Google Colab notebook. Now you are supposed to click here. The notebook will be connected in a while. Okay, this is a brief introduction of mine, and this is the brief introduction of this tutorial. And here, if you come here without watching this video, so the link of this video will be here. Okay, these are the contents. First, we will perform some sequence acquisition quickly, fetching sequences. Then we will perform some basic operations, some guided RNA design, and then we will identify the PEM sites and we will design the gRNA. Then we will perform some off-target consideration and then we will perform some efficiency analysis. It will be performed quickly. Now we are going to install BioPython. Just click here and the BioPython will be installed quickly. It may take 30 to 40 seconds. Okay, now I have the BioPython version 1.85 installed. You can clear this output as you like. Okay, now this is the biological context. I will use SAL1 gene from H. vulgare, or also called barley. So if you click here, let's say NCBI, and let me show you first here, select the nucleotide and I write SAL1. So we are interested to fetch a specific gene that is SAL1 and from at a specific or from the specific organism. So let's see. My internet is lagging. Okay. Now I have the first one, Hodium vulgare. It's actually barley. And this is the SAL1 gene. It's 2977 base pair of linear DNA. So you don't need to open this. Otherwise, if you want, you can. So we will just copy accession number cleanly from here and click here, show code. Now, if you down scroll here, you will see and just paste your sequence of gene here and click run. Once you click run, then you must see or you need to see the information of your gene accession number and some description lines, and then the base pair information. You need to verify those base pair information from here 2977. It will help you to validate, like, yes, the system has picked a similar gene. And here you can see the first 100 nucleotides of your genes. If you want to see more, just you can see here first 100 nucleotides. So you can make it, let's say, first 10 nucleotides, then you will see only the 10 nucleotides. If you want to see the whole genome, just write 2977 here and you will be able to see the whole gene, not genome. the whole gene sequence of SOL1 here. Okay, now we are interested to perform some basic operations. Just click here and it will retrieve 3 to 4 basic operations. The first operation, it will give you the RNA sequence. Once again, first 100 base pair are shown here. If you are interested to show more, then click here and check this 100 to that value you are interested in. So you will see the RNA sequence of that from the DNA sequence. Okay, this here you will see the protein sequence of first 50 amino acid, because I mentioned here first 50 amino acids. Okay, and if you are interested in reverse complement, so you can see here it has generated the reverse complement for first 100 uh, for the whole gene, but you are seeing first 100 nucleotides. Okay, and finally it prints the GC content for the whole gene. In this case, it is 43.80, and a kind of pie diagram for the A percentage, T, G, and C percentage. Okay, now we are moving towards the for the identification of PAM sites. I already discussed in the starting slides that what are the importance of PAM sites. Still, you can read this short biological context in PAM mechanics. So, uh, just click this one and you will see PAM sites, the representation of PAM sites on the whole gene. So, and here it just prints the first 5 PAM sites, but in total, this command found 103 PAM sites, and specifically it found NGG PAM sites. So let me tell you one thing more. It depends upon the system. If you are using a bacterial system, so the PAM sites will be different. If you are using an eukaryotic system, the PAM sites will be different. So if you are running this notebook, then you have to change. If you are, let's say, you are interested in CGG, let's say let's say NCC. N means any nucleotide. So in that case, it found 132 PAM sites on different locations. But in first case, we use NGG, so that's why that were 103. Okay, and if you want to see more identified PAM sites, so you can see here, like this command says, this line says that print 5. So let's say I'm interested in printing 10 PAM sites of the identified. So you can see from site 1 to site 10. So it's up to you how many positions or number you want to see, and this is just a visual representation that will tell you like where are your PAM sites are located. Why? Because if you are interested in designing a gRNA, like in next step we will design a gRNA, so it is essential like there must be a PAM sites, because it is the PAM site which will give a recognition sequence like I'm here to the nuclease enzyme for cutting. So now once again for designing gRNA, here is a small biological context. So you can read it now. Just click here and it has designed top 5. It has designed all the gRNAs for 132 PAM sites, but the top 5 are shown here. Once again, you can tweak the code and change the for the top 5 PAM sites. You just change this to 10, and then also change this to 10, and then uh, the code will be able to show you the 10 gRNA or the guided RNAs. Okay, this is the gRNA sequence. In this code, I have already used the scaffold sequence, which is normally a conserved sequence, and we keep it with the gRNA. So if you are not interested in the scaffold sequence, then you can uh, turn off this block of this piece of code by just using this sign, the hash tag sign. So automatically it will be blocked, or it will be the program will not count it. Okay, so these are your top five gRNAs. This is just a schematic figure, like how many RNAs you are seeing. So you are seeing the top five candidates. Now we are coming towards the off-target consideration. What is off-target consideration? For example, you have designed a gRNA. So is it sure, are you sure, like your sequence, the gRNA sequence will bind to that location you are interested in? So if not, that will be an off target. So it's just like that. For example, you design a gRNA or a guide RNA for the CRISPR for the location of 120, let's say a hypothetical location 120 nucleotide. So in this case, if your RNA is also matching to the position of 200 in the downstream, in that case, you will think that the gRNA will bind to the template DNA on 120, but no, it will bind to the 200 location. And the nuclease will go there and may perform a cut there if there is a PAM site. So in that case, you will destroy your the whole. I would say the whole experiment. You will perform some cutting or editing, whatever you are doing, but it will be off target. So that's why it is important to perform the off target analysis for your gRNAs. But most importantly, how you will perform. If you see the code, here I have used this thing, fetch the Hodium vulgare sequence, this one, genome record this. So let me show you what kind of record is this one. Here, this was a single gene we selected. Now I'm showing you like what I have fetched here. So in future, when you are performing the off-target analysis, it will help you to this step will help you how to perform the off-target analysis. Now you can see this is it says H. vulgare subspecies unplaced genomic scaffold pseudomolecule assembly whole genome shotgun sequence. So if you read here, I placed the whole genome shotgun sequence, the accession number of whole genome shotgun sequence for this species, no thanks, for this species, and I placed that session number here. Let's suppose you are doing some CRISPR things or some CRISPR analysis, or you are designing some CRISPR gRNA for human gene. In that case, you will provide the human genome sequence accession number here, because uh, in that case you will want that whether this gRNA I'm designing, it will attach some other places in the genome or not. So in this case, I put the whole genome sequence of this organism. In your case, you have to put your own sequence here for the whole genome. Okay, so let's perform the off-target analysis. and it says off-target analysis for the first gRNA, this one, fetch sequence this, the length of the whole genome was 66,000 something base pair, searching and found zero potential off-target sites with smaller than three mismatches. So that's a good thing. But if you are seeing some off-target in hints here, that means that it's not a good thing, or it's not a good result. You should consider your second gRNA or third gRNA or fourth gRNA. Okay, so let me clear it now. We are interested to calculate the efficiency of the target, the efficiency or the target efficiency of your gRNA you design. So you need to click here and it will tell you for the first five gRNAs, like the first one, that from position 20, the GC content are 55% and the efficiency is 80. For the second one, the GC count is 45, the efficiency is again 80, and like that. Okay. Now, what are the factors that determines the efficiency rule? So first one is the GC content. If the GC improves Cas9 binding without forming stable RNA structures. Because if in your experiment a stable RNA structure is formed, it will be harder for Cas9 to make a cut. 3' in avoiding GG at the spacers end enhances the efficiency and complexity. Diverse sequence reduce off-target risk and improved specificity. So that's it. I hope you enjoy my work. And one thing more, there are so many web servers. One of them is Chop Chop. One of them is Autumn AUTM, and so many more. The gRNA designer. You can also perform these steps on the those servers, and I would also suggest, if you are involved in the CRISPR experiment, so once you perform here, also you can validate your result on those servers, and let's see how it goes. So I hope you will enjoy my work. If yes, so if you are new here, please click here and subscribe to this channel and share with your peers and colleagues the bioinformatics insights. Also, you can share this channel and give me thumbs up. And uh, if you need any suggestions, or if you want to give me any suggestions, I read all the comment sections, so please drop your suggestion in the comment section. Thank you so much. Bye bye.